Well, welcome to Bodega Marine Lab. I'm John Lager, I'm Director of the Lab and Professor of Oceanography, and it gives me great pleasure to welcome you all here today. The Bodega Marine Laboratory was established in the 1960s, so do the math, that's quite a while ago. It's part of the Coastal and Marine Sciences Institute at UC Davis, and this lab, this place is home to quite a substantial research community, including almost 100 staff, researchers, faculty, students who are based here in Bodega Bay, but also many colleagues who are based in Davis. So our community extends beyond this place to Davis and beyond that as well. The research, the remarkable research and education that happens here is really based on three primary things. Probably the first one is sort of critical aspect of our facility is a seawater system. So we reach out to the ocean, bring the ocean inside, pump it through the building to many research aquaria, and this is paired with our access to field sites that this reserve and this location gives us. So we reach out to the ocean, right? We bring it in, but we also go to it. And that pairing of approaches is very powerful. I mean, we've been here a long time. We is sort of the corporate we, a broader community, since people have been doing sort of modern Western science research here since, the 40s. Lab is in the 60s, and so in a time of changing environment, changing climate, we have this sort of unique view, and we've become a coastal observatory during changing times. The impact of BML extends quite well beyond Sonoma County, and it's greatly enhanced by collaborators and visiting researchers, no more than those here who are here long-term, colleagues that come back year after year studying organisms and studying this place. Similarly, using the facilities that the lab and the reserve provide. They likewise develop exciting new insights and groundbreaking work. And a great example is Dr. Don Michaels, who's been associated, I don't want to say how long, Don, 50 years. Well, he did his PhD here and has come back repeatedly every year. He's established the crab lab and Colorado State University in the 80s, and has continued to intertwine his work on crab and integrative biology with researchers here. So special to have Don and Vicki Michaels with us today. You might recognize the name of the lecture. It gives me great pleasure to welcome everybody to this annual community lecture, the Dr. Donald L. Michaels Distinguished Lecture Series. There's a nice formal name for it. It's a celebration of science and an exposition of the impact of research in a changing world. The Michaels Distinguished Lecture brings us together to welcome into our community a leading researcher who illustrates how integrative approaches to biology can be used to address questions that span levels of organization, from within an organism to across organisms, within a system, across evolutionary time in many different ways, scales and time and space that biology is organized. The award honors the recipient in a way that advances our academic community, as well as establishing enduring links with the researcher. I guess our hope is somebody like Don who will continue to collaborate, I'm looking at you, Linda, continue to collaborate with us for another 50 years, and thus enriching our community as well, sort of synergistic way. This lecture always reminds me of the immense value we derive from being together in person. The last few days, there's sort of labs been abuzz with interactions between researchers and students and people from different backgrounds across disciplines and across origins where they're sitting, as I say, including the undergraduate class that's here during the summer and graduate students who are resident here around. So finally, in terms of introducing the lecture series, the Dr. Donald L. Michaels Distinguished Lecture Series was established through a generous gift from Vicki and Christopher Michaels in honor of Don and his career. And we look forward to many more of these, and we welcome you to join us in supporting the lecture series if you feel so inclined. Now it's my pleasure to introduce the 2025 Michaels Distinguished Lecturer, Dr. Linda Wegley Kelly from UC San Diego, and specifically from Scripps Institution of Oceanography, where she's an associate researcher in the Marine Biology Research Division. I'm not going to give a long introduction, because Linda can tell you more about herself, probably more succinctly and with more insight than I can. But I will say that she's an environmental microbiologist, so obviously studying microbes, and her research targets the critical role that these microbes play in nutrient recycling and organic matter transformation across coral reef ecosystems, seaweed aquaculture, and intertidal habitats, many different places. In particular, Dr. Kelly leverages marine biogeochemistry with microbial genomics, untargeted metabolomics. I can't say these words. Yeah. Who made up these words? Yeah. These complicated approaches to understanding how organic substrates shape microbial communities and ecological functions. So already you've seen, like, from microbes to, like, ecological functions and communities and all the different scales of organization. More broadly, I say she's a leader in opening our eyes to the immense, diverse, and wondrous ocean microbiome, right? Not just the gut, but in the ocean. Everywhere there's all these microbes doing incredible work, and we're just starting to learn about them. So please enjoy the lecture. Stay for the reception. Everybody's welcome. That includes you, whoever you are. We hope you stay and talk with each other and talk with Linda and other researchers here. Now it's an honor to welcome you, Linda, and I speak forward in saying that we're looking forward to your lecture, and take it away from here. You mic'd up? Yeah.
I think so. Can you all hear me in the back? All right. Thank you all for coming. It's been such a pleasure to be here these past few days. Everyone's been so kind. I love how beautiful it is here. I just like watching the grass, watching the deer in the coastal prairies has just been so peaceful for me. I needed to get out of the city. So thank you for coming and listening about my research. And today I'm going to talk about some of the applied work that we do in coral reef ecosystems. We are trying to, all restoration is hard, but we are trying to make advances for more successful outcomes. And so we're making some progress, and I still have a lot of faith. So I'm going to do the acknowledgements up front because I want you all to see that I was brought into this group by engineers at the University of Illinois, and they wanted to work with a microbiologist. And so they are materials engineers. That's Amy and Rosa. Rosa's a biomineralization expert. Gabe is a fluid mechanics engineer. Like, everything they do blows my mind. Joaquin's a postdoc who worked on the project. Kristen Marhaver lives in Curacao and works at Carmody. She is an expert in coral reproduction, and she's an incredible wealth of knowledge, just working so hard on the ground every day to protect these ecosystems. Zach and Emily are my graduate students, and Jean Yu is a graduate student in Illinois. And Forrest Rower was also part of this project you'll see at the end, and he was my PhD advisor. He's been telling me lies for over 20 years, but he's been a great part of my life. Okay, and I'm going to take you on the reef, about a minute long, and I'm not going to talk really through the whole thing, but I want to bring you to the reef, and I just want you to see what it looks like and the amount of structure that we have. Our Acropora palmata. You don't want to miss these guys. And so these are large area imagery that we take on the reef, and we go back to these sites every year and image the reef. And so you're kind of seeing these 2 years. And the reason the colors look so different is just because we take like thousands of photos kind of in a lawnmower fashion. And the difference in colors is just like the brightness of the day. So if there's clouds out, you can't really mask the colors. But we can use these three-dimensional models to estimate coral growth. It provides us this historic context for every colony. We can see which colonies are resistant to diseases and to warming, and we can track them over time. So it's really powerful. But one of the things I really just want you to see is just all of those shapes and all of those structures. The coral reef as a whole needs all that. Once you start losing all of that rugosity, there's a lot of implications for that. You're getting a flattening of the reef. So we lose colonies to warming events and diseases, and we need to do things to help keep them growing and keep all of that, all of those three-dimensional structures there. So this is just to highlight a little bit of the invisible things that are happening on the reef. I know the Zoom people can't see me when I step aside, but so the thing, I love this illustration. I work with an artist and he makes these beautiful illustrations, but what? Yes. Can I bend it? Is that better? Alyssa? Fine? Okay. So the one thing I just want to say, the only thing I don't love about this picture is that just here on the right, I want to see, it still kind of looks tall, even though we have these drawings of the fleshy algae. What's not true is when you really get a phase shift with the fleshy algae is that it's not tall anymore. You have this flattening. And so there's a lot of things that happen when you lose the structure, you lose the height of the reef. But some of the invisible things that happen on the reef is that you have much higher microbial biomass when you're in this state. So we do tend to see more diseases. It's just more microbes in the water. You get less recycling of nutrients and all of the functionality of the reef that happens. And you even can change the flow from where when you have more structure, you get more turbulent flow. You get better exchange of materials between the water and the benthos, and you lose some of that when you're getting this flattened reef. And these are all things that you can't see with your eyes when we're having reef degradation. So that reef, I'm not going to show you what happened to that reef in 2023, but it doesn't look like that anymore. We lost a lot of our brain corals, for example, to the stony tissue loss disease, and the warming's been really hard. So we're working on ways so that we can just kind of give the reefs a lift and help rebuild them when they endure these stressors. And so there's been a lot of different types of restoration. There's a lot of groups out there. There are challenges in restoration and some limitations. Sometimes there's not a lot of consideration about where you're building these nurseries or where you're putting them, what type of environment you're trying to outplan in. We also have there's a lot of work doing these microfragmentation and with branching colonies, and that's also wonderful. You can rebuild calcifiers on the reef, but we do also need to protect genetic diversity out there, so we do need to do sexual propagation as well. And with that, you're starting with larvae, and so that can be slow. You're not starting with, an adult colony that can grow really fast. For the first couple of years, they grow pretty slow. And then what we're also seeing is that even when we can produce baby corals in the lab, we're having pretty low success, survival, and outcomes out on the reef. And so we really want to enhance this by thinking a lot about where we're putting the juvenile corals, what types of substrates we're putting them on, and can we use substrates that'll actually give them a boost. And so just like a little introduction, and that's going to be a common theme in this talk, is that there's this pink, this is Christus coralline algae, and if you're from a region by the sea, you see this pink algae all over the place. But baby corals really like to settle and begin their life on these pink calcifying algae. It tends to be a great place for them to get a start. Rather than, regions like this, like these kind of like biophile, like fuzzy regions are not good places for these really small corals to get a start. So this illustration is just the kind of the coral life cycle, the coral reproduction life cycle. And so all of the kind of the arrows that you see that are kind of blue and green up here, like we've gotten really good at understanding the life cycle of corals, for some species of corals, figuring out when they're going to spawn. And we can do in vitro fertilization in the lab very easily. We're getting very good at keeping the larvae happy and alive, kind of over here. But where there's a few things, so settlement can be hard. They don't like to settle on anything. So you got to give them certain chemical cues. We are starting to get pretty good at that, but if you don't give them the right cues, they still won't settle on your substrates. But the biggest part is all down here, is just, once they settle and they become, and they're juvenile corals, keeping them happy and promoting their growth and outplanting them on the reef, this is all the parts that we're struggling with right now. So in my lab, we use approaches in microbiology and biogeochemistry and trying to improve coral restoration with the knowledge that we have. We use a lot of molecular techniques to characterize molecules and microbes in the marine environment. So some of the questions that we're answering in this context are, so can we understand like the establishment of the microbiome in juvenile corals to help them be more robust on the reef and be more resistant to disease? Can we engineer better restoration structures to improve outcomes? And how can we utilize chemical ecology to influence larval settlement and also just enhance growth and survival overall? So one of the techniques we use that we harness in my lab is that we can all benthic primary producers, whether they're corals or seaweeds, as they're photosynthesizing, they release a proportion of that photosynthesis into the surrounding seawater. So as they're growing, they're building biomass, but then at the same time, they're just kind of releasing extra molecules that they don't need into the surrounding environment. And those are all like just across the board, just very different kinds of molecules of all types. There's many different types of lipids and proteins and amino acids and even vitamins. And so we've been characterizing the types of molecules that different marine primary producers make and exude into the surrounding seawater. So one of the things that just to give you kind of just at the broad level is that each type of organism makes very different things. And so as you can see this in this Venn diagram, it's just all I want you to see is that these are different organisms. Struggling to see. So these are Poeciloptera and Porites. These are both corals. Porites is kind of more of a mounding coral, like a bommy, and Poeciloptera is more of a branching coral. They're both very common across the Pacific and the Atlantic. Turf algae is just like what you see on the rocks. It's kind of more like encrusting fleshy algae. Dictyota is a small brown algae, and this is CCA, the custos coralline algae. And then they all make like a different profile. Oh, so sorry, this is what I want you to see. All of these numbers are all unique molecules made by each one. And this in the middle is just 3 molecules that they all made. So not very many cosmopolitan molecules. And so this is just kind of a picture of a profile of everything they make. And you can even kind of see like that. So the corals, you know, they're still more similar than like the brown algae. Awesome, right? And then even CCA, the calcified algae, is kind of in the middle, so a little bit more similar than the fleshy algae. Turf is an assemblage of browns and reds, so that's nice. And so this is just to show that we can just see the different things that organisms make. And this is a picture of everything that they've made that we can measure on the mass spectrometer. And every color was made by one of the individual organisms. So orange is CCA. We're going to focus on CCA today because this whole talk's about CCA, Crestus coralline algae. And so all of these molecules right here in dark and just shown here in orange, they're only made by CCA. And so the question is that we know that corals like to settle on CCA. So can we like extract the yummy molecules that CCA make and then use them in restoration? And that's what we try to do. So this is like just to show you again is that these is just when things get biofouled in the marine environment. Anyone who's like coexists with the marine environment knows that. Get like really fuzzy things on everything that you have. And these are baby corals. They're really tiny. They don't want to get smothered by all this. And they like to grow on something like this. It's just not fuzzy. And so what we want to do is we want to keep the substrates clean, and we want to promote, and we want them to be interested in our clean substrates. And then we also hope that those substrates will help them grow faster and have better survival outcomes. This is Zach. He was an outstanding PhD student, and he went down and he extracted the molecules that CCA makes. This is CCA in a glass beaker. And so then we can extract all those molecules. And then he did settlement experiments where he just added them to the water. So we didn't embed them in any substrates or anything. We just squirted them into the water. And so to see if we could kind of elicit that response with the CCA, just kind of in a little, like a petri dish. So this is, you can see that if you just have water, they don't settle at all. We had like 0 settlement. And then the tissue derived, that just means we kind of ground up the CCA and just added it to the water. And 2 different species of CCA, you do get an increase in settlement, but we also could induce interest in settling just with the exudates. So that was like the first step. And so this is just kind of a diagram to show you this, just kind of like what I mentioned in words is that and it doesn't hurt the CCA, it does when we grind them up, but these ones, when we do the exudates, we just leave them in the beaker under a light cycle for about like 6 to 8 hours, and then we can put them back in the aquarium and reuse them. This is a non-invasive technique. But we can use these different types of resins, and they bind the molecules that are suspended in the water to the resin. Then we can extract them back out, and we can put them in the freezer so we can keep them. And so you can't, these resins, they don't absolutely capture 100% of all the molecules, but they capture a good proportion of things that are there. So it's not the whole community. So Zach had to do a test where he said, does my extracted CCA still induce settlement at the same rate as whole CCA? So he did this. And so everything in blue is just the controls. We have low settlements. And then this is the whole non-fractionated CCA, so the whole thing. They did pretty well. And then these are the different fractionated. So this is like a 0.1x, so not very concentrated. Equal concentration to here, but fractionated. And then in red was like 10x concentrated. So we can get that response again from the materials that we can preserve and keep in the freezer and use them for engineering. So we go to our engineering friends. And in this case, in this example, we are working with a researcher at SIO. This part of the project wasn't done with the University of Illinois folks. But Dr. Wong Prasert in his lab, he does a lot of, he's like a nano engineer. He does all of this. He can grow symbionts in polymers and keep them growing. He does all this amazing engineering work. And they have designed this, he calls it BioInk. It's like it's a liquid that you can add like a set of molecules to, and you can actually just kind of smear it on any substrate, and then you put it under UV light, and it kind of like, it cures it. So then it's like, it like hardens it. And then those materials, like underwater over time, it's like a slow release. So then we can have substrates that have this BioInk kind of smothered on it like an icing. And then over time, they'll release the CCA molecules into the environment. And so the hope is that we can just put these out and get better sediment on substrates that we've designed that have other properties that could be beneficial. And so Samapti her paper just got published earlier this spring, and they did a really amazing job. It's a really great paper. Okay. And so we're gonna move to just talking about a little bit about how, like, can we use the microbiome or can we just push the microbiome a little bit of these juvenile corals to be more robust, to be more resistant to diseases and pathogens, and just really, we just want to keep them alive. And so one idea is that you can expose the juveniles who are here, you can see that right here, they don't really, they start out with very few symbionts, both bacterial and algal. So that you can see, you can't even really see them. They're translucent because they've not developed their algal symbionts like they are here. And so if you can see here, this is like a beautiful picture that Emily took, where you can see all of their algal symbionts inside. And at this stage, we also anticipate that they're establishing their own microbiome of bacterial symbionts. And so Emily asked the question is, can we generate specific biofilms that are common on the reef that promotes the establishment of different microbiomes with these juvenile corals? And just to, it's just to reiterate a little bit, but this is just kind of a separate, a distinct study that we have, is that I showed you a little bit just to say that the CCA does induce settlement, but our partners, our fluid dynamic engineer partners, can actually show that the exposure to larvae with the CCA exudates actually completely changes the behavior of the larvae. They gravitate towards it, as that you can see here. So this is like in a little tiny baby flume, and everything that's white is a larvae, and they're just kind of equally dispersed throughout the little baby flume. And then what they do is they put the exudate right here at the beginning, and after a couple minutes, the larvae, like, they start slowing down, and they start all like gravitating over to here to where the CCA signal is. So they really do cue in on something about those molecules, and we hypothesized that, and these are things that we still need further exploration, but that there's a reason why that happens evolutionarily, and that these should be thought of as kind of happy homes for them, right? That this is going to be an environment where they're going to thrive, and that there's something about the CCA that helps promote survival. Certainly, they think it smells good. So Emily set up these different aquaria and she kind of established these substrates with the engendering different biofilms for about 2 months. And so these substrates were, so there was a control aquaria and just had seawater flow through seawater. Then she had an aquaria where she had CCA in it, and that made these ones. See all the pink. And so we're getting both like representation of the CCA from being exposed to it, but we're also getting a biofilm of microbes on the substrates that grows in response to the CCA exudates. And then she tried Dictyota, which we would think is more of an antagonist on the reef, not a great place for a baby coral to settle in like a patch of Dictyota. But so she was still interested in seeing the response. And so here is the Dictyota kind of enriched substrate environment. And here you can kind of see more of this kind of, maybe over here you can see it better, these like lighter green, which is probably like little baby Dictyota. So we did do a gentle clean of them before we settled the larvae, because kind of this got kind of fuzzy kind of diatoms and whatnot, it would not be great. So we used kind of this little, like a silicone like mascara wand. It was very gentle. We didn't want to disrupt the biofilm, but we did need to get rid of kind of some of the scuzzy stuff. But you can see the controls are pretty white. The CCA is starting to look like CCA, and then the Dictyota treatment is kind of a bit of a mix. And this is what they look like under blue light. And so these are the trays that we do the settlement in. They're just clamshells. And afterwards at the reception, Emily's a much better person to answer your questions. I'll introduce her later. But so when you look at them under the blue light, I mean, they're very striking. You can see all the different primary producers on them. And this is the control. And these green dots, splotches, are the settled larvae. So those are the baby corals. The baby corals, even though they were conditioned for 2 months in seawater, the corals did not love the control substrates. Here's CCA. These like orange ones are kind of the beginnings of the CCA, but all these greens are baby corals, and they actually really loved the Dictyota treatment as well, which was confounding a little bit to us, but that's how science works, not what we expected. So this is just to show the data as just kind of in bar charts, is that controls did not much. CCA, some settlement, and quite a bit of settlement on the Dictyota, and it was also more variable. They had like a lot of like gregarious settlement. On certain tiles, there'd be like 50, and some tiles there wouldn't be very many. But we, and by we I mean Emily, this is her brainchild, was mostly interested in understanding which microbes that we could find that that the baby corals are establishing in response to the different treatments. Right? And so we're trying to understand how neighborhoods can influence the microbiomes of the baby corals. And she's had to do, this is a summary of, and I just want you to see that like we do see some differences, is that all of these microbes here, they were all enriched in the CCA treatment, as they were the most abundant. And Emily had to do a lot of work just to try to look at a signal to noise because coral larvae are really small. And as we have shown you, they settled on a biofilm, which is like a thick mat of microbes. And so for every sample, she takes another sample of the biofilm next to it, and then she does all of these calculations to look for a signal of microbes that are in the coral and not in the biofilm next to it. And so these are only microbes that pass that test. And you can see that we are seeing that we have some specific types of some specific taxa that look like they were engendered by their surroundings. Okay. So the really hard part about creating corals in the lab and putting them back in the sea is what happens to them when you put them out. When you put them back out on the reef, they over and over again, we just find that if we generally settle, we put them on something. We put them on the sea floor where they came from. And that is not working very well. We're getting very, 0 to 10% max survival rates when we're doing these outplants out onto the reef. And so we are looking for ways that we can get corals to grow better and to have better outcomes, and then potentially move them down to the bottom later. And so these coral reef arcs were developed by Enforced Rowers Lab. And so there's been a big team of people, not just included here, but primarily I want to talk about Jason Baer. He did his entire PhD on these arcs, and he has about, I have some of the publications, but he has about 5 papers on these arcs. We're really just trying to create an environment where we can have better survival of the outplants, at least for a while. And so the idea is that we would just get them off the seafloor, one, but also that we would just put out these are what we call arms that are settlement plates where you can get a lot of the biodiversity of a coral reef growing on them. And then that you could move these arms to the arcs, and that you'd kind of create this kind of oasis. So that's the idea of the arcs just themselves, not really using them in a restoration framework. But we'd also like to utilize the arcs to promote the survival of our outplants. What they look like, they are these geodesic structures are really strong, and so they ended up being like a good shape that they weren't susceptible to breaking on the reef. They're made out of PVC. They're not very expensive. We can build them, but it's just so Forrest is a phage biologist. He's a virologist, and so it just so happens that they are the shape of a phage, and so that's not a coincidence at all. But phage are also bacteriophage are also very tough, so maybe there's something to that. But so this is a picture of one of the arcs that was deployed in Curacao underwater. And so we have been using them, and they've had a lot of success with the arcs. They're very strong. The The weakest point that we find is the tether point. That's like really the only problem that we've had with them, is that sometimes they can get dragged and if that is not strong enough. They increase the oxygen levels, which helps the organisms grow. We've seen huge recruitment of fish and invertebrates that are help kind of maintaining the systems and keeping them clean. And they actually accrete a lot of biomass. They've got really heavy, really fast in some places, like Puerto Rico. And so these are some of Jason's papers. He worked for about 5 or 6 years on these and did a lot of work. And so that's another cool